Yeah, hi everyone. I'm Ira. I'm a research engineer at DeepMind. But what I'm here to talk to you about today is a work I did during my PhD at Imperial College London and the biomedical image analysis group. So let me start with an outline of the talk. So first of all, I'm going to introduce some basic concepts from network science and graph theory. And then I'm going to briefly talk about analogies between Euclidean and irregular domains. Some brief overview of spectral and spatial graph convolutions and what the differences between the two are. And for this three points, like apologies to the ones of you that are already familiar with the concepts. I know that the tutorials yesterday covered some of this material. I just wanted to make sure that we're all on the same page for the rest of the talk. When it comes to applications, I'm going to cover metric learning, population graph-based classification, class activation mapping, and parcellations, and all of this in the context of medical images. Networks or graphs in mathematical terms are objects that are used to model pairwise relations between data points. And one thing that makes them so great and like very relevant in a wide range of applications is because they offer a global view of data structures. And they allow us to gain insights about the data and extract global meaningful patterns, while at the same time, they can help us increase performance in certain tasks, like, for example, classification tasks, by leveraging pairwise similarities between the elements that serve as network nodes. Apart from that, there are certain tasks that are only designed on networks. For example, if we wanted to do some kind of community detection in social networks, then we would have to use graph representations. An important distinction between classes of graphs is natural versus constructed graphs. Natural graphs refer to networks that arise in social, biological, communication, or transportation networks and contexts. And they refer to data that inherently lie on a graph structure. So in those situations, we don't have to specify the graph structure ourselves, but it's inherent in the data. One example that you can see on the left-hand side are brain networks, and in particular, structural brain networks. So this is an image of a connectome, which I'm going to talk more about in the next few slides, which captures the white matter tracts connecting different brain regions, while the color codes are indicating the directionality of these connections. And on the right-hand side, we have a transportation network. In the case of constructed graphs, we have only the data available, like this data might be lying on a manifold potentially, and we want to construct a graph that is meaningful enough to represent the whole set of data points. And this is often computationally expensive. It's quadratic to the number of elements or nodes that we want to capture in the graph. And at the same time, there's no universal recipe, but only common good practices. And one example of that is a 3D mesh, in this case of the heart, that is very often used in cardiac applications in medical imaging, but also in other graphics problems. And kind of a very basic definition, and again, apologies if this is too trivial. But yeah, in order to fully define a graph, we first need a set of vertices or nodes, and then the number of nodes, we refer to that as the cardinality of the graph. We also have the set of edges that tell us how the different nodes in this graph are connected, along with a similarity or adjacency matrix. And if all the entries in this adjacency matrix are binary, we have a binary graph. Whereas if they're real numbers, then we have the case of generic graphs. And another important distinction is between undirected and directed networks, where in the former case, the adjacency matrix is symmetric, whereas in the directed networks, the adjacency matrix is asymmetric. Now, as I mentioned earlier, this talk is mostly going to be about connectomics and neuroimaging. And this is what I spent pretty much 3 years of my life investigating. And so I've been looking at the brain from a network perspective. And why is that even interesting? Is because cognition itself is a network phenomenon. So all the complex functions that we can perform arise from interactions between the individual neural connections in the brain. At the same time, there are certain disorders, neuropsychiatric and neurodevelopmental, for which there's barely any physical trace, and there's only changes in the wiring or the strength of the connections. But how do we get from a standard 3D MRI image to a network representation? First of all, we need to extract the cortical surface. And by cortical surface, we refer to the gray matter, so the neuronal bodies at the surface of the brain, where most of the computation is performed. Now, this cortical mesh is actually a very fine-grained representation. So it can reach up to hundreds of thousands of vertices. And we want to find a way to process these meshes in a more kind of computationally efficient way, but also in a way that allows us to draw meaningful conclusions. That's why there's a big area of research that is trying to parcellate the brain. And by parcellation, we essentially mean a clustering problem, where we try to group neurons or areas on this cortical surface into meaningful and functionally coherent regions. And this can either be anatomically inspired or it can be function inspired. And after we've done this step, then we obtain the imaging data, which can be either diffusion MRI data, where we're looking at to track white matter trends, or neurophysiological data, which we quite often capture with functional MRI data, that are an indication, that give an indication of how blood oxygen levels change in certain areas of the brain, which is indicative of the brain activity in those areas. So in the former case, by counting potentially the number of streamlines connecting to brain regions, and in the latter case, by looking at the correlations between the time series that we obtain with functional MRI across time, we can construct this structural or functional brain networks that are represented, that can be represented with an adjacency matrix. And finally, by modeling each brain parcel as a node and each entry in these adjacency matrices as an edge, we can obtain this network representation that allows us to use traditional network analysis and graph theoretical measures to draw conclusions about certain disorders. And there's actually a big field of research in neuroscience that is looking at graph theoretical measures and how they differ in between healthy controls and individuals with schizophrenia or epilepsy. Now, I guess many of you are familiar with the successes of in computer vision in the Euclidean space with ImageNet and very like being able to perform very complex tasks with standard images. But given that this talk is mostly about irregular domains, I think it's very important to understand the analogies between the two. So in traditional medical images or 2D images, we have a regular pixel or voxel grid that provides the domain structure. That corresponds to the graph structure in the irregular domain. We also have a variable number of neighbors per node and no specific node ordering. Whereas in the Euclidean case, we have a fixed number of neighbors per pixel, and we have an intrinsic ordering. So we know which pixel is left, right, up or down. Now the image intensities from the Euclidean domain correspond to the node feature vectors in the irregular domain, while the tasks that we can perform with graphs are basically there's two main tasks, or let's say the equivalent ones from the Euclidean domain are image classification for graph classification. So this is trying to classify a whole graph, whereas the equivalent of an image segmentation task is a node classification task. Now, some elements from spectral graph theory are actually provide the foundations for the field of geometric deep learning. And spectral graph theory allows us to analyze data on top of a network or graph representation. And in this example, we have a graph signal X that is defined on an unweighted, on an undirected weighted graph. So this signal corresponds to the blue bars. And the Laplacian matrix is the most powerful operator in spectral graph theory. And that allows us to analyze and process networks. But it also acts as a diffusion operator, which means that if we have a heat source on a particular node, then by multiplying the signal with the Laplacian matrix, which is a degree matrix minus the adjacency matrix, then we can see how the signal, this heat source diffuses across future time steps. And graph Fourier transform, which is the analogous of the traditional Fourier transform in the Euclidean domain, allows us to expand this signal X in terms of the eigenfunctions of the Laplace operator. And here's one great example from Javier Bresson's course that helps us understand the analogies between the the Euclidean Laplacian and the standard Fourier basis. So essentially, after decomposing the Laplacian into a set of eigenvalues and eigenvectors, we can obtain the modes of variation that correspond to the set of eigenvectors, which you can see visualized at the bottom in increasing order of eigenvalues. So as the corresponding eigenvalues increase, then we can see sharper transitions in the values of the eigenvectors for neighboring nodes in the graph. Whereas for the lowest frequency component, we can see that all nodes essentially have the same value. So overall, geometric deep learning started by trying to reinvent building blocks of convolutional neural networks for irregular domains. Now the field is spanning a wide range of applications and problems that could not be handled with Euclidean approaches. But there's two main streams of work. In spatial filtering, we try to generalize the notion of the patch of pixels. And these provide a more natural analogy between with regular structures, as well as provide a more intuitive interpretation of the filters. One downside with these approaches is that they require defining a neighborhood system and a node ordering. In spectral filtering approaches, the convolutions in the spatial domain correspond to multiplications in the Fourier domain. And we don't need to go through the tedious work of defining a neighborhood or a node ordering, which some early works did. While at the same time, they provide a principal definition of the convolution operator, with the downside that they're not transferable between different structures, because as we said, they're parameterized on the Laplacian. So if the Laplacian changes, then the filters are no longer transferable. Now, by parameterizing the filters directly on the Laplacian and the eigenvalues, we obtain very computationally expensive formulation, because we need to do the eigen decomposition, which is quadratic complexity. And we also obtain filters that are not localized. And that's what inspired the early works of Deferard and others, which I think you covered yesterday, as well as Kipf and Welling with the kernel trick, and later on Levy and Al with Cayley nets. These polynomial parameterizations ensured lower computation complexity, and at the same time filters that are strictly K localized, with K being the order of the polynomials. So here, when we say K localized, we mean that if we have a second order polynomial, then we're only filtering information or incorporating information that is at most 2 hops away from the central node. Now, during the early stages of my PhD, I was really interested in trying to estimate the similarity between a pair of graphs, as this was very useful for several downstream tasks, and had to rely on more traditional graph theoretical approaches or graph kernels. I use graph edit distance methods that are sometimes hard to scale, but at the same time, they involve some heuristics in order to tune parameters and things like that. And when graph convolutions, convolutional networks came out, it seemed like a very natural framework to try out and obtain such a metric of similarity between a pair of graphs. So in this work from 2017, what we did is that we obtained labeled graphs for a set of individuals. So for each individual's brain, we use the fMRI time series and the correlations between them to construct a connectivity matrix. And then by representing each brain region as a node and using the connectivity information as node features, we had a labeled graph per individual. Now, as I mentioned earlier, we had to ensure, in order to process these networks with spectral graph convolutional filters, we had to ensure that the graph structure is preserved across all the samples. And that's why we obtained a population level connectivity of the graphs. Now, as I mentioned, the goal of the task was to estimate a similarity metric between a pair of graphs, given only their classes. So the labels that we had available are these two graphs. Do they belong to individuals from the same class, where class can be disease status, or it can be a certain phenotype, like for example gender. And based on this information, we would try to force graphs that belong to the same class to have higher similarity, and graphs that belong to different classes to have lower similarity scores. And we processed pairs of graphs with the Siamese graph convolutional network, which means that the weights were shared between the two branches of that were processing each of these input connectivity graphs. Now, a dot product layer essentially provided a similarity estimate of the latent node representations after the C layers of convolutions. And then the fully connected layer allowed us to combine the information from the different nodes to obtain this final similarity estimate. Now, we tried this approach on two different data sets. The first data set came from UK Biobank, which is a large prospective study planning to acquire imaging data for a large number of healthy subjects, predominantly healthy subjects. So this data set is pretty homogeneous, and it's also pretty large for neuroimaging standards. We used 2,500 subjects and their resting state fMRI data. By resting state, we mean that the subjects were not asked to perform any task, but they were imaged at rest. And our data set was relatively balanced, given that we framed a gender classification task there. We also used the Abide dataset, which was quite different in the sense that it was data acquired from 20 different acquisition sites, and therefore it was highly heterogeneous. And the task in that case was to classify individuals as healthy controls or patients with autism spectrum disorder, and as these have been diagnosed with cognitive assessment. And again, we used resting state fMRI data, and this time for a smaller group of subjects, 871. Again, relatively balanced data. As I mentioned, this multi-site setup was quite challenging, and also there were several sites that were overrepresented in the data set. So we wanted to make sure that our experimental setup accommodates for that and avoids any biases towards a particular imaging site. Therefore, we stratified both the training and testing sets in terms of the acquisition site. And in order to obtain the training pairs, because the Siamese network was trained on pairs, we constructed a bipartite graph just to make sure that each individual graph was not seen more often than others. Whereas in the testing set, we used all combinations, all pairs between individuals in the test set. So one of the downstream tasks that we used this similarity estimates for was K-nearest network classification. And here the green bars reflect the accuracy achieved on the UK Biobank data by experimenting with 3 different loss functions. The first loss function was the standard hinge loss, The global loss, its objective was to maximize the distance between the means of the same class versus different classes similarity estimates, while at the same time minimizing the variance for each class of network pairs. While the constrained variance loss, because we observed that in certain cases the global loss would cause the similarity estimates to collapse around the means, we wanted to allow we wanted to only penalize the variance above a certain threshold, and this is what seemed to lead to better performance compared to the global loss that had been previously used in Siamese architectures for image patches. We compared this like graph-based learning approach to 3 different baselines. One that didn't use the graph structure at all, which is the Euclidean. Essentially, we estimated, we only incorporated the connectivity matrices as if they were traditional feature vectors. We looked at random walk kernels that capture the graph information, but there's no learning component. And the large margin nearest neighbor classifier that does not use the graph structure again, but the distances are then learned using the classes of the samples. Another downstream task that we used this learned similarities for was manifold learning. And in this slide, you can see the 2D projections obtained with locally linear embedding based solely on the similarity matrix between the pairs, all the testing pairs. So on the left-hand side, we have the baseline, the large margin nearest neighbor classifier. Whereas on the right-hand side, we have our proposed approach. And top and bottom are the 2 different data sets. The stars then indicate the center of mass for the 2 classes in each task. And even though this is less obvious in UK Biobank, in the Abide dataset, we can see that this approach was able to separate, even though they're still quite mixed, it was able to separate in this 2D manifold subjects, healthy controls from patients with autism spectrum disorder better than the baseline. However, while performing this study, what we could observe and what was quite striking was that the similarity between brain connectivity networks was heavily influenced by the acquisition side. So the first 2 panels of plots here show intra-site differences for controls, patients, and individuals from different classes. And you can see that we can pretty easily distinguish between the 3 categories. However, as soon as we merge all the sides together, which is what we see on the right-hand side of this slide, then it's really hard to distinguish between networks coming from the same class versus networks coming from different classes. So what you see here plotted is a distance between pairs of networks. So what we wanted to do was to be able to robustly classify disease from healthy individuals based on their imaging information, while at the same time using non-imaging or phenotypic data, like for example, acquisition site, gender, age, or other information that is often available along with the imaging data. And in order to do that, we use the population graph to perform semi-supervised classification. And by semi-supervised, we mean that we had a population graph in which a node represented an individual, in contrary to the previous approach where a node was a brain region and we had a different graph per individual. So here we have all individuals in a single population graph. The feature vectors for each node correspond to the imaging data, while the edges capture imaging and non-imaging similarities between these individuals. And the semi-supervised element comes in through the fact that the feature vectors of all samples are observed at training time, while only the labels of the training data are used for the optimization procedure. Now, some design consideration during this effort were, first of all, deciding on the distance metric. Would we use Euclidean distance? We could use cosine distance, correlation, and that's between the feature vectors of the nodes. Or if we have like distributions, data distributions on the nodes, we could use a distance from information theory like Kullback-Leibler divergence. In terms of the data features, we could use the original features or handcrafted features, but we could also use learned features like using principal component analysis, sparse coding, non-negative matrix factorization, or autoencoders. And unfortunately, there's no single solution that fits all the problems. So we had to try out a few of those. Now, overall, as I mentioned earlier, we had a single population graph, which was processed by a series of L hidden graph convolutional layers. And at the output layer, each node would have a feature vector of the same dimensionality as the number of classes we were trying to predict. And after the softmax layer, we would obtain the class with the highest probability of representing this individual, and optimized the network using the standard cross-entropy laws. The population graph's adjacency matrix was estimating using both the similarity between the feature vectors, which is the first element in this formulation, while the second element is reflecting the agreement between non-imaging measures, and this is what we mean with the function here. The data sets that we evaluated this approach on were the ADNI dataset, which is a large cohort of healthy individuals and individuals with mild cognitive impairment, as well as Alzheimer's disease. And the task that we wanted to perform was predict the conversion from MCI to Alzheimer's disease, which is much harder than predicting the healthy controls and whether they're going to convert to Alzheimer's disease in the future. There's more subtle differences between these two groups. So that included more than 1,600 longitudinal samples, about half of which corresponded to 289 subjects. So we had multiple samples from the same subjects. So half of them were diagnosed as with Alzheimer's disease at any follow-up time. The node information, so the node features, corresponded to the volumes of 138 segmented anatomical brain regions, while the edge information corresponded to captured the age, gender, and longitudinal acquisition information. And the latter means that samples from the same subject at different points in time would be connected with high edge weights, indicating that there is a strong connection between these two samples. And we again used the Abide dataset for autism spectrum disorder diagnosis, where the node information corresponded to vectorized functional MRI networks. And the edge information captured the gender, the acquisition side, which, as I mentioned earlier, was pretty important, as well as the correlation between fMRI connectivity matrices. So one aspect that was pretty important was the feature selection strategy. And here, as you can see, for example, in the case of the autoencoder, the latent representation would be the one that defines the learned input features for a population graph. So it was kind of, the feature selection was a pre-processing step before feeding the population graph and its node features into the graph convolutional network. So all this analysis was purely performed on the training data. We don't touch the test data here. And we compared multilinear perceptron, so essentially projecting the input features to lower dimensional output from the NLP. Autoencoders, which try to reconstruct the input by having this learned lower dimensional representation. Recursive feature elimination, which is more kind of traditional approach, and then principal component analysis. And we could see with the ROC area under curve that recursive feature elimination led to the best performance in this setup. We also experimented with a degree of the polynomial, and this corresponds to the polynomial, the spectral filters. And the results that we got were consistent with other works in the literature that show that, especially with ShebNet, a polynomial degree of 3 or 4 leads to the best performance, and increasing the degree any further would actually lead to saturation. So we wouldn't get any improvement, or maybe the results would actually deteriorate. Now, the different colors indicate different graph structures for the population graph. So we wanted to make sure that this similarity graph that captures phenotypic information and imaging information and similarities between subjects was actually meaningful and useful for the classification task. So this phenotypic graph is the one that you can see in kind of dark pink color. And we compared that to a random graph that has the same edge weights, but with edges randomly shuffled within the graph. And 4 different KNN graphs. So here the KNN graph would be constructed solely based on the feature vectors of the node, so disregarding the phenotypic information. And the complete graph would connect every sample to every other sample. So we're essentially agnostic to the similarities between subjects, while all would also add the similarity information on the complete graph. So it's pretty obvious that the phenotypic graph is actually contributing by a lot to the classification task, since it yielded almost a 10% improvement in classification accuracy for the ADNI dataset. We also looked at, again, this is for ADNI, we looked at different phenotypic measures to use for constructing the population graph. And what stands out is that adding the APOE4 allele, which has been associated with Alzheimer's disease. So we essentially connected individuals that both have the allele. And this actually seemed to improve performance as well compared to leaving that out. Another thing that we experimented with was whether averaging the output of networks using different seeds or ensembling would lead to better performance. And we saw that overall ensembling was sometimes better, but for the top performing graph structure, it didn't really make any difference. Now, some follow-up work from Technical University of Munich experimented with, built on top of this work, and proposed the so-called Inception GCN, which is inspired from work presented in 2015 at CVPR with the Inception net. And what they proposed to do was leverage spectral convolutions with different kernel sizes and use that for disease classification with the population graph. So it's exactly the same setting that I just described, capturing at each inception module, capturing different receptive fields, essentially. And this work that was presented at the IPMI conference in 2019 did exactly that. Another thing that they experimented with was the effect of the kernel size or the polynomial degree. The authors claim that consecutive layers do not need to have the same receptive field as we did in our work. And the plot that you can see at the bottom actually demonstrates that. So the different panels correspond to different affinity matrices, essentially, for the population graph. And then on the y-axis, we have the polynomial degree for the first layer, while in the x-axis, we have the polynomial degree for the second layer. And then the heat map indicates the classification accuracy for each combination of kernel sizes. And what they observed was that overall, having a higher receptive field in the first layer versus the receptive field in the second layer led to better results. So it's better to, if early layers are capturing more kind of global information, while the later layers, subsequent layers, would capture more fine-grained information. And this is kind of consistent to the computer vision literature, where we see that in the early layers, it's where kind of more high-level features are picked up, whereas in subsequent layers, we have more fine-grained filters. They also experimented with different aggregators to merge the outputs of the inception modules, which included concatenation and max pooling. And their disease prediction results summarized here are showing that even though they could get an improvement in classification accuracy on the ADNI dataset, about 2 points, that was not the case for the Abide dataset, which again, so you can see that the Inception GCN does not outperform the baseline, which again highlights how challenging this data set is, and how what we actually need is a better way to perform domain adaptation and transfer the knowledge that we gain from one site to another different site. Now, another work that we did back in 2018 was, and given that we're in a medical context, we don't care that much if we have a super high prediction accuracy, but we don't know where it's coming from. What I mean by that is that if we're able to classify between individuals with a particular neuropsychiatric disorder with high accuracy, we want to be able to know what are the underlying brain structures involved in these differences, so that then treatments can target these brain areas. Or even better, if we can understand the mechanisms that give rise to these differences, then it's even better for treatment. So this is what we attempted to do with this setup, that essentially introduced the global average pooling after the layers of graph convolutions. And this global average pooling would essentially project each filter map from the last convolutional layer to a single neuron, and then a fully connected layer would yield the the two-dimensional output for the classification for the logits, essentially. So what we could do then is multiply the subject-specific weights with the filter maps of the last layer, and that would yield a class activation map for each individual subject. And the next step involves identifying the top K activated brain regions, or nodes in this case, across all subjects. And by summarizing this information in a single map, we can then see which are the brain areas that essentially allow us to perform this classification with a certain accuracy, which means that these areas are the ones that introduce the differences between the two classes that we're comparing. And we applied this for sex classification on UK Biobank data to obtain these graph saliency maps, essentially, that allow us to interpret and visualize the learned filter maps. And the outcome was, it highlighted the differences in the default mode network, which is a network that is activated at rest when we are not performing a particular task, and which has been shown in previous neuroscience literature to be different between males and females. So it's pointing towards a direction where we can actually use such techniques to identify the brain areas that are contributing to class differences. Another stream of work that was performed later on at the University of Montreal and was published at the Medical Image Analysis Journal had to do with cortical parcellation. So as I mentioned in one of the early slides, parcellation is supposed to provide functionally coherent brain regions and enable downstream tasks like a meaningful brain network analysis. And even if traditionally it's supposed to be kind of unsupervised, equivalent to a clustering task, the authors in this work, they used it in a supervised setting. So they had a ground truth parcellation, and they wanted to train a model that allows to learn the same parcellation for unseen individuals. And the way they did that was having a graph for each individual subject that corresponded to the cortical mesh. And the node features in that scenario included the spectral coordinates and the so-called depth for each node on this cortical mesh. And because spectral convolutions rely on keeping the graph structure fixed, they had to make sure that they register all cortical surfaces to a single reference, with spectral matching and the Procrustes algorithm. And here you can see how their method performs to different baseline, either in Euclidean or the spectral domain. And it's pretty obvious that the Euclidean domain is not the right the right domain to perform such a task, because the brain surface is so convoluted. It doesn't help to use the original Euclidean coordinates of the cortical vertices. But what is also obvious is that having a Markov random field on top of as a post-processing step would further improve their results, which were impressively higher, better than FreeSurfer, which is a widely used software in cortical surface analysis. Now, most of this talk has covered applications of spectral neural networks, but I wanted to also mention a spatial approach, the graph attention networks that were proposed by Velikovic in 2018 at ICLR. And their formulation is computationally efficient and essentially promises to be to give strictly localized filters that also tailor the importance of the weights for the different nodes using this attention mechanism, which, if you're familiar with transformers in NLP, is kind of like a similar idea. So essentially what that entails is computing these coefficients E that capture the similarity between the current hidden representations of a pair of nodes. And then these coefficients are normalized with the softmax-like function to obtain attention weights for each pair of nodes. And then this information would be aggregated either using concatenation or averaging to yield the hidden representation of a node at the next time step. This approach was again applied on the parcellation task using the Human Connectome project data, which is a data set of about 1,000 individuals that has been released. It's a research data set. And the graph attention networks focused on Broca's area, the parcellation of Broca's area, which is the area of the brain that is responsible for speech production. And they compared this approach both to graph convolutional networks, so the spectral approach, as well as baselines that completely disregard the graph structure, like node average, for example, or node MLP. Now, in terms of future directions, I would like to highlight reproducibility, especially in neuroimaging and neuroscience applications. It's extremely important to be able to reproduce findings, either in terms of brain regions involved or mechanisms involved in certain disorders, which is quite often not rewarded enough in neuroscience conferences, but there's definitely things have improved over the last few years. And second major point is domain adaptation. So conclusions that we make based on a single cohort, being able to translate those in a new cohort with data acquired from a different imaging site and at the same time being able to leverage information drawn and conclusions drawn at these different imaging sites. So I think that there's a lot of room for improvement there. And the other point that is really important is interpretability. So being able to understand if we're getting very good performance, why are we getting this performance? What are the underlying mechanisms? And because I think these 3 points are just extremely important in neuroimaging and neuroscience applications, this is why I wanted to have them like duplicate these items here. Another very interesting future direction is spatiotemporal modeling, because so far we assumed that the graph structures are static, which is more often, it's quite often actually not the case, especially with functional MRI data. Even if we're saying that we're capturing the brain at rest, it's pretty unlikely that the 30 minutes or 15 minutes of an imaging acquisition, which is one of the longest ones actually, is representative of the individual's functional connectivity across the lifespan. And here I'd like to share some useful resources for in case people want to dig deeper into this topic. And I would like to highlight at the bottom actually a very interesting series of blog posts by Michael Bronstein that is providing some very valuable insights into the field. And if you're interested in medical imaging in particular, there's the GRAIL workshop that is organized as a satellite event of the largest medical imaging conference, and is held for the 3rd year this year, and is featuring speakers that are amongst the pioneers in the field. Yeah, so that's all for me. And let me know if you have any questions.